We are Estonia-TUIT iGEM team from the University of Tartu, Estonia, and we present our project, BeeYeast, yeast engineering to fight bee viral infections. Honeybees play a vital role in human life. As key pollinators of both wild and cultivated crops, they are indispensable in maintaining ecological balance, food security, and the global economy. They produce valuable products that we use, such as honey, beeswax, propolis, bee venom, and others. Bees make a significant contribution to the global economy, accounting for a substantial portion of food production, estimated to range from 235 to 577 billion US dollars annually. Unfortunately, the population of honeybees worldwide is declining due to climate change, excessive chemical use, and infectious diseases. The deformed wing virus, or DWV, is a major bee-infecting virus that causes millions of bees' deaths every year. The issue is worsened by external parasites known as varroa mites, which infest larvae, pupae, and even adult bees, transmitting DWV and compromising their immune systems. It can pose a significant threat to pollinator populations and agricultural ecosystems. Currently, several strategies are used to fight viral infections in beehives. However, the existing methods are not always sufficient to suppress DWV infection and also might negatively affect the health of the bees. Therefore, there is a need for innovative approaches to safeguard bee populations. In nature, bees use RNA interference to defend themselves against RNA viruses. How does it work? Well, RNA interference is a process where small pieces of RNA can shut down protein translation by binding to the messenger RNAs that code for those proteins. When a virus infects a bee, the viral genome is replicated in the bee cell, leading to the production of double-stranded RNA, or dsRNA. The honeybee's dicer-like enzyme recognizes and cleaves this dsRNA into shorter 21 to 22 base pair molecules known as short interfering RNAs, or siRNAs. These siRNAs are then captured by organoid, a component of the RNA-induced silencing complex, or RISC. Subsequently, one siRNA strand is released, and the other, called the guide strand, directs RISC to complementary viral RNA. This process leads to the cleavage of viral RNA, halting viral replication and protein production. Unfortunately, the natural source of siRNA needed for defense mechanism is viral RNA itself. It is produced only after infection of bee cells, making it insufficient to defend bees against viruses. Our team aims to exploit engineered yeast to enhance honeybees' natural RNA interference response, helping them to fight deformed wing virus infection. For that, we modified yeast cells to produce short interfering RNAs, or siRNAs for short, which can block the expression of bee pathogen genes. First, we screened the deformed wing virus genome to select for the best candidate sequences, which can be used by the RNA interference machinery. Then we cloned these sequences into a yeast plasmid vector to express short hairpin RNAs in yeast. In order to produce active siRNAs from the precursor, shRNAs, dicer protein must cleave the hairpin of shRNAs. Since Baker's yeast naturally lacks RNA interference machinery, we introduced the gene encoding for dicer protein into S. cerevisiae. To deliver the synthesized siRNAs to bees, we plan to feed them with pills made out of our genetically engineered yeast. Bees would absorb siRNAs from the midgut and accumulate it inside the cells. There, these siRNAs would bind to endogenous organoid and RISC complex, providing an additional level of protection against viral infections. To strategically plan and optimize our project, we employed modeling techniques for computational simulation of siRNA production in yeast, along with the steps that happen in bee cells. This allowed us to determine the initial experiment setup and evaluate the performance of the proposed approach in relation to desired final result. Moreover, modeling siRNA production in yeast allows to optimize various aspects of the process, including the choice of promoters for shRNA and dicer expression, potential concentration, efficiency of shRNA folding, and its stability. we divided our project into 4 stages, enabling us to model each stage individually and conduct a comprehensive evaluation to ensure its success. Modeling of shRNA production revealed differences between shRNAs and showed that the folding process is not a bottleneck in shRNA accumulation. During the subsequent phase, it was predicted that dicer expression controlled by PTEF1 promoter is sufficient to cleave most of the synthesized shRNA, converting it to siRNA. Previous studies have found that delivering synthetic siRNAs to bees reduced DWV-related mortality, indicating that the small RNAs are capable of passing through the digestive system while retaining activity. By modeling siRNA uptake by bee cells, we found that siRNA concentration in the lower nanomolar range is sufficient for effective RNA interference, while higher concentrations are more prone to cause off-target effects. And finally, the modeling of RNA interference in bees showed that the continuous supply of optimal siRNAs is likely to have a strong beneficial effect on bee antiviral defense and supports the setup of our system with engineered yeast-producing, ready-to-use siRNA rather than shRNA that would require further processing by bees. Testing the effectiveness of siRNAs in suppressing deformed wing virus infection in bees would be a labor-intensive, costly, and complicated process, mainly due to the involvement of working with live virus culture. Therefore, we designed a reporter system in yeast Saccharomyces cerevisiae to assess the impact of these siRNAs. Since S. cerevisiae does not possess RNAi machinery in the initial phase, we introduced dicer and argonaute proteins, which are essential elements for RNAi from a closely related yeast species, Saccharomyces castelli, into the cells of S. cerevisiae. This resulted in the engineered yeast strain capable of enzymatically cleaving shRNA into siRNA molecules. At the next step, we created a plasmid containing an shRNA sequence under the control of the pGAL1 promoter. This plasmid is responsible for expressing specific shRNA molecules that can target particular regions of deformed wing virus RNA. To facilitate a quantitative readout of the efficiency of siRNAs against deformed wing virus, we created third component of our system, a reporter consisting of the gene coding for green fluorescent protein fused to the viral target sequence in its 3' untranslated region, right after GFP stop codon. The reporter expression, as well as shRNA production, is controlled by an inducible pGAL1 promoter. The system works the following way. When yeast cells contain all 3 necessary components and are growing in glucose, argonaute and dicer proteins are produced, but neither GFP target nor siRNA are expressed, as GAL1 promoter is repressed. This leads to the low background level of GFP fluorescence. If we grow cells in media containing galactose instead of glucose, both GFP reporter and siRNA are expressed, along with dicer and argonaute. In this case, the GFP fluorescence level depends on the efficiency of RNA interference. If expressed siRNA is efficient in targeting the region of viral genome fused to GFP coding sequence, the RISC complex will degrade the GFP target transcript, leading to low GFP fluorescence. However, in the case of non-efficient siRNA, the reported transcript is not degraded and GFP signal is still high. By utilizing this system, we aim to assess the degree of inhibition exhibited by the siRNAs against complementary viral RNA derived from the DWV genome. The system can be used to search for the best siRNA candidates against DWV, and potentially can be expanded for the screening of efficient target sequences from other viral pathogens. To test the efficiency of the designed siRNAs using our yeast, we used flow cytometry. We measured the GFP fluorescence in yeast cultures 24 hours after inducing the expression of shRNA and the GFP target reporter. In control strains where the expression of both GFP and shRNA was repressed by glucose, the fluorescence was very low, serving as the background control. In strains expressing dicer and argonaute proteins with induced expression of GFP target and shRNA, the observed GFP fluorescence levels were also low, comparable with the background fluorescence level seen in the control strain. This indicates that our designed shRNAs are able to completely suppress the GFP reporter expression, making them strong candidates as anti-deformed wing virus agents. In contrast, yeast cells expressing dicer Argonaut, the GFP target sequence, but an shRNA designed against a different target sequence exhibited high GFP intensity. This finding underscores high specificity of the interaction between siRNA and the target sequence. In another control strain lacking the shRNA plasmid but containing dicer, Argonaut, and the GFP target sequence, the fluorescence levels were high as expected. Furthermore, a strain lacking RNAi machinery proteins but capable of producing GFP target and shRNA showed elevated levels of fluorescence, confirming that the observed reduction in fluorescence was indeed a result of the RNAi response. The results of our experiments lead to 3 important conclusions. First, the presented work validates the published results that introducing dicer and Argonaut proteins to S. cerevisiae is sufficient to constitute RNAi response. The experiments showed that viral sequences introduced to the 3' UTR of a GFP-coning transcript can be targeted by siRNAs in yeast, making this a simple way to test the efficiency of different siRNAs. We observed complete suppression on GFP reporter expression with tested anti-DWV siRNAs, confirming that they are strong candidates for further studies. In the future, we aim to improve the yeast-based siRNA efficacy testing system by placing GFP target and siRNA transcriptional units under the control of different promoters. Test the effective siRNAs against the full-length genome of deformed wing virus. Investigate the uptake of siRNAs by bees, and test the putative negative side effects of siRNAs in bees. During the development and execution of our iGEM project, we dedicated significant attention to adhering to safety regulations. We conducted experiments using yeast and did not use whole genomes of the deformed wing virus to keep our work at the biosafety level 1. To ensure our iGEM project aligns with responsible practices and real-world needs, we undertook several key human practice activities to shape our project. We explored apiculture regulations in Estonia and consulted with experts from the Agriculture and Food Board. We reached out to 10 beekeepers from 4 countries to gather insights on disease treatments and assess stakeholder interest in our product. Our findings and specialists' feedback confirmed that the deformed wing virus is widespread in Estonia and highlighted the need for novel treatments against viral infections. Encouragingly, some beekeepers expressed keen interest in our novel approach once it is proven to be safe. We also partnered with experts in the fields of virology, bee biology, RNAi research, mathematics, and incorporated the received insights and feedback into different parts of the project: conceptualization, design, safety, modeling, and education. To fill the gap between science and society, our team organized and participated in already traditional public events, education fairs, and info sessions, hosted an international summer school, conducted workshops, organized the art exhibition, and an escape room. We developed new educational materials, board games, and online games, translated them to different languages, used them at public events, and made them available on our wiki. Our educational approaches aimed to not only be academically informative, but also easily accessible and entertaining for everyone, and include diverse communities into life sciences. During the design and implementation of our iGEM project, we partnered with various stakeholders and relevant experts and incorporated the insights and feedback into different stages and parts of the project. Engineered yeast to produce short interfering RNAs, siRNAs for short, which can block the expression of bee pathogen genes. Developed a novel siRNA efficacy testing method in yeast and used it to validate siRNAs that may block the deformed wing virus propagation in bee cells. Designed educational materials, hosted international summer school, and conducted multiple public engagement events. Developed multidisciplinary, hard and soft skills, and expanded our network. And just as importantly, we shared many emotional moments and became friends for life. We want to thank everyone who contributed to the project, external experts, sponsors, and our Science and Technology family. The project would not be possible without the team and all the people who helped us during the season.